Hello everyone, and welcome to Chapter 1 of our textbook for this semester. So in this very first chapter of microbiology, what we're really going to be discussing are some of the background information on what is microbe, how did we even get to this discipline. So on the first slide we have here, we're going to talk about the following things. We'll talk about the early years of microbiology. We'll talk about the golden age of microbiology, where we were really seeing a lot of advancements and a lot of developments in the discipline. And we'll also talk about the modern age of microbiology. Where is microbiology going now, and what does the future hold? I like this first chapter because it's really a nice layout of the way that we're going to go through a lot of the topics in our text. And it also gives you a basis of reference on why we understand microbiology in the way that we do and what we can do with this understanding. Now, I want you all to pay attention because for my class, these learning objectives, these are going to be your quote-unquote study guide. This is the information with the accompanying pages in your book that you'll want to make sure that you specifically pay attention to. So, what does life really look like? Now, if we take into consideration that prior to the 19th century, there were no way for us to look at microbes. We actually thought that life just arose spontaneously. We didn't have the microscope yet, so we didn't really have a true understanding of microbiological life, or that there could be organisms that were smaller than the naked eye. Antonio van Leeuwenhoek was a Dutch merchant who actually studied biology and science. And really there wasn't a discipline called microbiology at the time, but he studied the science and biology. He studied it as a hobby. He didn't do it for money, and there was no such thing as being a microbiology teacher. That wasn't a viable career option at the time. But he began making these simple microscopes where he was able to look at specimens. His simple microscopes are very much like the microscopes that we use today. There's a lens. There was a place where they would put the specimen, and you could move that specimen either closer to the lens or further away, and you could move it left or you could move it right. And much the same way we do with our compound microscopes. Now, his microscopes were very simple, and we don't say they were simple simply because they were rudimentary. We say they were simple because it only had one lens. We have a compound microscope because there are two sets of objectives. We have the objectives that are down on the nose of the microscope, and then we also have the ocular pieces. And we've discussed that in lab. Often he made new microscopes for each specimen that he made, and he would have these really fancy viewing parties. And they would look at their teeth scrapings, they would look at their poop even at these really fancy parties just to see this microbiological life. There's even one fancy aristocrat that's known to say that when she looked into her teeth, she saw that there were all these microorganisms and it was just teeming with life. And she said, "Egad, there's a zoo in my teeth." So there was a very nostalgic sort of a way that we think about microscopes when we talk about Anton van Leeuwenhoek. And there he is, our Dutch merchant with his microscope. And this is a replica of what his microscope looked like. So this is, we have our lens, and let me pull out my pointer here so you guys can see what I'm talking about. So we have the lens here, and then we have the needle which would hold the specimen, and then you have the ability with this screw to move that needle either closer to the specimen or further away, or you can move this needle here and move the specimen up or down. So after the 19th century, after Anton van Leeuwenhoek actually had his microscope, and then during that time we saw scientists like Pasteur, Louis Pasteur, which we'll talk about quite a bit. We saw physicians like Semmelweis and many other early notable microbiologists that we were able to sort of think of them as not just being these vital forces that, you know, spirits just come from, but that they were an actual classification of life. So from that point, we needed to find a way to classify them. So Carlos Linnaeus is the person who's credited with developing a taxonomic system for naming plants and animals and how to group them together. So with Leeuwenhoek's microscope, we looked and we discovered, or we decided that life didn't just consist of plants and animals, but there were also other types of life that need to be classified as well. We had bacteria, archaea, fungi, protozoans, algae, and small multicellular animals. Now, these are probably the kingdoms that you remember learning as a little kid, or the kingdoms that I remember learning when I was in college and I was in high school. We now take those kingdoms and we've kind of reclassified it a little bit into 3 domains, and we'll talk about that later. So we look at the prokaryotic cells because that's what microbes are. Many microbes, I shouldn't say all of them, but most microbes, a good chunk of them, at least the ones that we're going to discuss in this class, can be classified as bacteria. They're prokaryotic cells. Prokaryotic cells are going to be single-celled organisms. They're unicellular. They don't have a nuclei. They're much smaller than the eukaryotic cells that make up your cells. If you've ever done the cheek cell lab, then you remember looking at your cheek cells and notice how big your cheek cells were in comparison to the little tiny bacteria that was found on the inside of them. But bacteria aren't the only prokaryotic cells. We also have archaea. Archaea are small unicellular cells that are that reproduce asexually, and at first we used to think archaea were very much like bacteria because they were small and prokaryotic, but we found that there are a lot more similarities between archaea and the domain of eukarya. So some notable differences between archaea and bacteria. Bacteria have a cell wall that's made out of this special starch polymer called peptidoglycan, or actually it's a polysaccharide polymer called peptidoglycan, which is a protein-sugar combination. And then for the archaea, they're actually made out of polymers that more closely resemble starch than peptidoglycan. So peptidoglycan is a very important word because we're going to talk about that throughout the semester. Peptidoglycan is a component of bacterial cell walls, whereas archaea, their cell walls aren't composed of that same protein-sugar combination. So here we have a classic cheek cell, which we have stained in class if you are taking my class. And then we also see the bacteria that are hanging out in the cheek cell. So on to fungi. There are some fungi that would be considered microorganisms. Molds and yeast are 2 examples of that. Some notable features of fungi is that instead of being prokaryotic like the archaea and the bacteria are, they're considered eukaryotic. So they're going to be much bigger cells, kind of like your cheek cell was much bigger in comparison to those prokaryotic bacteria that were hanging out inside of it. Fungi must obtain their food from other organisms. They do have cell walls, but just like the archaea, the cell walls of fungi are different than the cell walls of bacteria. The cell walls of fungi are primarily made out of a polysaccharide called chitin. So it's a specialized carbohydrate called chitin. For your exam of Chapter 1, you should be familiar with all of the features of bacteria that we covered, bacteria and archaea on the previous slide, and also on this fungi slide and the subsequent slides that we just are going to talk about. You should be able to compare and contrast these different classes of organisms. So here we have spores and we have the budding cells of the eukaryotic fungi. Now we're on to protozoa. Protozoa are single-celled eukaryotes, so some similarities of things that we've already discussed. Bacteria and archaea were prokaryotic, fungi and now protozoan are eukaryotic. And remember, eukaryotic cells are bigger and much more complex than prokaryotic cells. Protozoans are a little bit more similar to animals in that their nutrient needs and their cellular structure. They don't have a cell wall like the fungi and the archaea and the bacteria. Animal cells don't have a cell wall either. Instead, their outer membrane consists of just a cell membrane or the plasma membrane. Some protozoans Most of them, in fact, reproduce asexually, while there are some that will do sexual reproduction, and they have various different means of locomotion. Some can use a pseudopod, like the amoeba, that it's considered a false foot. They just kind of blob their way through the environment. Some have cilia, like the cilia on your eukaryotic cells in your trachea, that will beat, look like kind of like little oars on a boat that are just beating in the same direction, and that will propel the organism through its environment. And then some have a flagella. The flagella on protozoan is going to be different than the flagella that we see on bacteria in the way that it moves. So there are fewer, there are stitches of the cell that are fewer, they're longer than the cilia, and they move in a whip-like fashion. Whereas for bacteria that are flagellated, that have flagella, they're going to move in a rotor. They're kind of going to spin in a circle. And when we get to chapter 4, we'll talk more about the specifics on the types of flagella that we'll see in prokaryotic cells. So here we have the amoeba that would move, and it's considered a type of protozoan. It moves via its pseudopods. And then we have the cilia here and this paramecium that would help to propel this organism through its environment. And you will also see the euglenas flagellum. And you probably had a chance to look at many of these organisms in an introductory biology class, and you'll also be looking at these organisms here in microbiology. Now on to algae. So far we've talked about bacteria and archaea. They're prokaryotic. They're the only prokaryotic cells that we've talked about so far. And then we talked about fungi. Fungi could be multicellular or it could be unicellular, and thus fit in the classification of microorganisms. Like bacteria and archaea, they also have cell walls. But unlike bacteria and archaea, their cell walls are made out of a different polymer. Then we went on to talk about protozoans and how protozoans are more like animal cells than any of the other previous organisms that we talked about, and they have different means of locomotion. And now we're on to algae. Algae can also be, like fungi, multicellular or unicellular. These are the only organisms that we have discussed thus far in this set of lecture notes that are capable of photosynthesis. As a result, most algae is green. Now, I'm going to stop and pause right there because there are some bacteria that are capable of photosynthesis. So that's why we said in that slide there are various nutrient needs of bacteria. Some of them are photosynthetic, some are oxygenic photosynthetic like algae, some are anoxygenic photosynthetic. Some can use things like petroleum for an energy source. So bacteria have a wide range of things that they can use to get energy. So there is that similarity between algae and bacteria, that they can some bacteria can undergo photosynthesis. Algae is characterized on the basis of its pigmentation and the composition of its cell wall. The cell wall in algae differs between the different types of algae we have. And here is the Gliocaspa, this is the spiral gyra that we're looking at here. And then these are various types of diatoms. So this is unicellular algae. And then the algae that you see in your pool or the algae that you find in the ocean, that would be a multicellular example of algae. Other types of organisms of importance to microbiologists are parasites and viruses. Parasites are multicellular animals, and viruses, well, we're kind of on the fence. There's some scientists that say viruses are living. They say they're living because they do have their own DNA or RNA or genetic material. And then there are some that say viruses are not living because they can't reproduce by themselves. Being able to reproduce and also having genetic material, those are 2 of the 7 hallmarks of life. Viruses don't have all of those hallmarks of life. So even today, there is still a wide debate on whether or not to classify viruses as living or non-living. And we'll talk in much more detail about each of these different classes of microorganisms as we go throughout our semester. So here we have a parasitic worm, and remember it is a large multicellular animal, and then these are the red blood cells, and you can actually see that this worm is able to engulf some of those red blood cells, just to give you a picture of size. And then we have here a virus that is infecting a bacterium. And it's really kind of cool. It kind of looks like, you know, one of the alien invader ships that you'd watch on Independence Day or something. But this is what we call a special type of virus that we call a phage or a bacteriophage. Bacteriophages are only going to infect bacteria. And the typical way that many of these phages work and many viruses that are non-phages work is that they will insert their genetic material into the bacteria, and then using the bacteria's own genetics, then it will make replicas or additional viruses. And when these new viruses are assembled, then they'll leave outside of this bacterial cell, killing the cell and then moving on to other cells to infect it. And we're going to also work with some bacteriophages later on in the semester. So now on to the golden age of microbiology. So scientists were really trying to answer these 4 questions that help to get us to this specific discipline of microbiology. 1, is spontaneous generation of microbial life possible? What that's really asking are vital forces or spirits or God or the gods responsible for bringing about life? You have to remember, prior to the 19th century, it was believed that if you were to get sick, or if food was to spoil, that we had to blame evil spirits for that. Even bad smells, evil spirits were blamed for that. So the question came about is whether or not spontaneous generation, life not coming from life, just from vital forces in the air, was responsible for the microbes that Anton von Leeuwenhoek was seeing in his Nancy Ewing party. The second question that we wanted to ask was, what causes fermentation? And at this point, they were really wanting to understand, when they talked about the term fermentation, why would food spoil? Number 3, what causes diseases? Once again, this kind of relates to that very first question, that it was once believed that if you got sick or you smelled bad, that God wasn't happy with you, and that's why you got sick. And that's where diseases came from. It came from your own bad thing that you've done to anger God or anger the universe. And then the 4th question is, well, if we can figure out what causes diseases, then is there a way for us to prevent infection and disease? So let's handle the first question first. Does microbial life spontaneously generate? Well, as I said before, some philosophers and some scientists thought the past thought that living things rose from 3 processes: asexual reproduction, sexual reproduction, and from non-living matter. It was Aristotle that we first credit with proposing spontaneous generation, that living things can arise from non-living matter. Now you must keep in mind that Aristotle didn't really have all the same tools that you have as a budding microbiologist or even as a microbiology student. So he was really just working with what he could see with his own eyes and the observations that he and other people of his day were making. So let's talk about Francisco Redi. Francisco Redi had an experiment because it was once believed that maggots came from decaying meat. So what he decided to do is that when he had his decaying meat, he kept it isolated from flies. And that decaying meat that was isolated from flies, well, it never actually developed any maggots. However, the meat that was left in the jar that was open to the flies, well, maggots and infestation soon arose. So as a result of Francisco Redi's experiment, scientists began to doubt Aristotle's theory that life could arise from non-living matter. And here we have this experiment because Redi believed that, well, once he saw that maggots turn into flies, he thought that, well, maybe the maggots are laying eggs on the meat. Now, you are very intimately acquainted with why this piece of meat didn't generate any maggots and this one did generate maggots, but you have to keep in mind that we're talking like 17th century, so this is really groundbreaking stuff we have here. So some people said that because he didn't allow any air to get into his meat, the vital forces that were in the air were unable to get to the meat and allow for the spontaneous generation to take place. So he did another experiment where he took the jar and he just covered it with a piece of cheesecloth or gauze and poked holes in it so that enough air would be able to get to the meat. And just as it can be expected, since the flies were not able to physically get to the meat, there were no maggots that arose from it. So Redi's experiments were the very first to prove biogenesis. So biogenesis and spontaneous generation are at opposite ends of the spectrum. Spontaneous generation says that life can come from non-living matter, and biogenesis says life can only come from life. So, of course, in the field of microbiology, we can't just have one person make a statement and one experiment and then the argument is over. Of course not. We have to have a debate about it. So John Needham made another experiment, and his experiment used beef gravy and infusions of plant material. So scientists were once again, they're on the fence. They were arguing about whether or not large animals could not arise spontaneously, but they thought perhaps microbes could. So Needham's experiment with beef gravy is actually in support of spontaneous generation. Spalzani came back and he had another experiment. He concluded that Needham, when he had his broth in there, the broth had microbial life in it. The broth was left open for Needham's experiment. And since the broth was left open, of course, it was exposed to the air and it became infected with microbes. So Needham said, we'll see, the vital forces are in the air and we can get microbes. Those really tiny organisms, they do come from non-living materials. So what Spalzani said is that Needham failed to heat the vial sufficiently to kill the microbes that were already probably in the test tube already. And that he also failed to recognize that microorganisms exist in the air and can contaminate the experiment. So spontaneous generation of microbes does not occur, which we know now, but it took a couple hundred years to actually become just basic scientific law. And that law that we have is all living things come from other living things. And you all have probably known this as the cell theory, that living things and living cells come from other living cells. So critics said that the seal cells that Spalzani had in his experiment, that they didn't allow enough air for the organisms to survive, and it destroyed the life force that was in there. So even at this point, after Spalzani was able to show the fault in Needham's experiment, after Francisco Redi did the experiments with the meat, there is still a debate as to whether or not microbial life is able to spontaneously generate itself. And then came along Louis Pasteur. Louis Pasteur designed flasks that had a swan-shaped or S-shaped flask. These are classic S-shaped flasks. And so what he did was that the flask was a very, very narrow tube. The flask with itself was nice and round and big. And then he would put broth in there, and he would heat the broth up. And then instead of just having a nice, wide, open mouth, he would pull out the glass so it made a nice S shape, and it was hollow so that air could still get into the flask itself. So this way, we wouldn't have the problem that Spalzani had with his critics saying that, well, not enough air is able to get to the broth, and as a result, that's why the microbes couldn't spontaneously generate. There was sufficient airflow, and he was heating the material in order to kill the microbes that are in there. So that when the flask was tilted, the dust from the bend in the neck would be swept into the flask, and then the microorganisms could be caught in both of the necks of the flask, and thus not contaminate his broth. So here we have our infusion that's boiled, so it's probably just like chicken broth or beef broth, and then here's the part where he'd pull out this flask, and it would be hollow so that steam could escape from it. Now notice that there are two bends in the flask. We have a bend here, and we have a bend here. So even if we allow it to sit out, air that wants to move into the flask is freely able to do so. However, there would be microorganisms could get caught in either one of these breaks, that the microbes would actually settle in the dust, and then they would never actually contaminate the solution. Now, this S-shaped or swan-necked flask and the ability to keep the broth or whatever it was free of contamination for months on end, this was revolutionary. This answered so many questions concerning the economy of the early 19th century, or the 19th century. One of which was, how do we get wine from one part of the world to another part of the world without it spoiling? With Louis Pasteur and his new process, which he coined pasteurization, we were able to figure out that problem. In fact, his design was so flawless that we still use pasteurization today to preserve many of our liquids and our wines and our cheeses and our milk and many other things in orange juice that are pasteurized. So what Louis Pasteur did to really come about it, to arrive at how he might develop these swan-shaped necks and swan-shaped flasks, and he didn't just come up with it overnight. He actually used the scientific method. Now, you guys are probably pretty familiar with the scientific method, so I'm not going to go over it in great detail, but he had an observation which led to a question. Questions are going to help you generate a hypothesis. So the question that Louis Pasteur had was, how can we keep microorganisms from contaminating the broth, or how can we keep microorganisms from fermenting our wine or spoiling our food? And so he had a hypothesis that maybe there's a way for us to keep it out, and we need to, that's the answer to this question. The hypothesis then has to be tested. So Louis Pasteur went through several different designs before he came up with one that actually worked effectively. And then your results are going to either prove or disprove your hypothesis. If you accept your hypothesis, then it will lead to a theory or law. Keep in mind that the leading to a theory or law does not happen with just one great experiment. We saw that with Francisco Redi, that there were other scientists that came and tried to debunk what his experiment said. So in order for something to be a theory or a law, it has to be proven over and over and over again using a variety of different experiments from a variety of different scientists or people that are conducting this experiment. If we disprove our hypothesis, then it's either rejected and say, that's just not the case, or we can go back to the drawing board and modify our hypothesis and go through the process over again. So here is a great schematum of the framework for the scientific method. So I'll let you all kind of look at this, and I do want you to make sure, because there will be questions on this for your exam, that you understand the steps in the road to get to that are taken for the scientific method. And to be honest, many of you guys really use the scientific method in your everyday troubleshooting when you're trying to figure out why your iPad won't work or why your printer won't print. So then on to our next question, what causes fermentation? Well, Pasteur really kind of answered that question for us, didn't he? So spoiled wine threatened the livelihood of many grape growers, and this was a problem for the economy, as I said before, that we could have wine that was, when it was shipped to the individuals that were receiving it, they didn't want to pay for it because it was spoiled. So unless you lived in a location that was near a vineyard, you didn't want to purchase the wine, which is going to cause a catastrophic economic decline for the grape growers. Some believe that air caused fermentation, and they were somewhat right because it's the microorganisms in the air that are causing fermentation. Others, such as Louis Pasteur, they believe that it was the microorganisms in the air or living organisms itself that caused it. Winemakers actually funded research of methods to promote the production of alcohol and prevent it from spoiling as a result of fermentation. If you heat wine or alcohol too much, then it will evaporate. So there had to be a middle ground there, and that's where Louis Pasteur and his idea of pasteurization came into place. So the debate over the cause of fermentation also linked with the debate over spontaneous generation. So in answering that question of how does life arise, we were also able to answer the second question of what causes fermentation and how to prevent it. And notice how the fermentation relates directly to Louis Pasteur's S-shaped flask. And we can also see how there are various different incarnations of this hypothesis as to what caused fermentation and the experiments that were used, and then the observations that were made, and whether or not hypotheses are going to be accepted or rejected. So as we said before, Pasteur's experiments led to the development of pasteurization. And what pasteurization is, this is something I want you to remember as a definition, is pasteurization is the process of heating liquids just enough to kill most of the bacteria. Now, the pasteurization that was used in the early 19th century is a lot more archaic than the pasteurization that we use today, but the principles and the basis behind it are still the same. We would heat our liquids up to a certain temperature for a certain period of time in order to kill most of the microorganisms that are in there so it doesn't spoil. Now, we do recognize that even though we use pasteurization, either it's ultra-pasteurization or flash pasteurization, that our milk will still spoil. And that's because it doesn't kill all the bacteria, and if you open up your milk and you use it, you're introducing even more bacteria to the milk. Then it doesn't kill all of the bacteria, but it kills most of it. So this pasteurization process became heavily used not only in the wine-making industry, but it became heavily used to manufacture all sorts of different things. And what we also realized is that some of those byproducts of fermentation, like acetic acid, which we use for vinegar, let's see, other citric acid, which we use for flavorings of things. If you're really into hot Cheetos and Takis like my children are, you'll see citric acid as one of the ingredients on there. It helps to give things a little spice. It's used for a flavoring of things. And even looking at how the ethanol, we use that for alcohol. So we have also been able to use some of the byproducts of fermentation for various different products. And here is a great little table that's found in your textbook that talks about some of the many industrial uses of microorganisms and how we get these different organisms and how we use them for different types of products. So the notable ones of interest are cheese, alcoholic beverages, soy sauce, actually all the food because I'm just into food. And then some other ones that are other products, antibiotics, pest controls, drain openers, and even some vitamins are isolated from bacteria. So bacteria kind of get a bad rap. They're not all bad. They don't all always just cause disease, but we've been able to find ways that they can be useful for our everyday lives. So more on what causes fermentation. Bucher's experiments demonstrated that fermentation does not require living cells. He showed that enzymes can promote chemical reactions, and we'll talk a lot more about enzymes when we get into bacterial growth. And his work was really kind of the foundation or some of the early work that was needed for biochemistry. Now, Louis Pasteur's work was more organismal in that he was concerned with just the bacteria themselves, whereas Bucher's work was looking at the different enzymes and different chemicals that are produced from these organisms. And these chemicals or these enzymes, these small proteins, couldn't even be seen with a microscope. So that makes what Bucher's experiments were and his understanding of biochemistry even more groundbreaking and phenomenal, in my opinion. Pasteur, not only did he solve the problem of how to get wine from Italy to England without it spoiling, but Louis Pasteur also developed the germ theory of disease, which basically says that germs or microorganisms were responsible for disease, not because you were poor, not because you were ugly, not because you did something bad, but because you were infected with microbes, that's what caused the disease. Robert Koch took that a little bit further, and he said that specific microorganisms cause specific diseases. And the way that he was able to arrive at that understanding is through what we know as Koch's postulates. And he was able to use them in understanding how anthrax, the bacteria anthrax, causes the disease anthrax. So with Koch's experiments, some other things that he was able to develop in his question of what causes diseases, Louis Pasteur got the ball rolling when he said microbes cause disease in his theory behind it. But Koch took it a little bit further and said, well, do all microbes cause the same disease? And the answer is no. Different microbes cause different diseases. Now, in order to arrive at this postulate, some things that he had to do was he had to be able to stain the bacteria to see them. We've done simple staining, and we'll do simple staining in lab. Simple staining just means that you're only using one dye to stain your specimen. He also took the first micrograph pictures or pictomicrograph pictures of bacteria, which is kind of like when you guys take your cell phones and then you put it up to the eyepiece on your microscope and then you take a picture of it. His was like that, but way less sophisticated. He also took the first photograph of bacteria and disease tissue. So once again, he's doing this investigative study using the scientific method to just demonstrate that the bacteria that are seen in the picture are the same bacteria that are seen in this disease tissue. So it must be this particular bacteria causing this disease. But he didn't just stop there. He would actually isolate the bacteria and grow it on media. So he used steam to sterilize growth media. We use an autoclave now. You don't have to use it. We have an amazing lab coordinator that does that for us. But all of the agar dishes that we use in lab, they've all been sterilized so that we know that there are no bacteria that are growing on there. Once again, this is a credit to Koch because he wanted to make sure that there were no other microorganisms competing with the microorganism of his interest as he determined what caused this disease. He also helped to develop these what we call aseptic lab techniques in order to transfer bacteria. Much of it we're going to do in our lab class, and much of it you'll do in your microbiology lab class as well. And then he was also one of the people to really support this idea of bacteria as a distinct species. So here's just a picture of some blood agar and the different types of colonies that you would find on that. Now, in our very first lab, we've actually taken some samples from the environment and we put them on those small agar plates. And what you'll notice is that not all the colonies on the agar plates are going to be exactly the same. In much the same way that the bacterium colonies, and these are called colonies, on this plate are not all exactly the same. In fact, they all have a different what we consider morphology. Some are small, some are glossy and raised, some are more of a milky color and they're not as glossy, they kind of look dried out. Some are stippled a bit, some have irregular edges, some have different colors. So those different colonies represent different species of bacteria. So each colony, as in bacteria number 5, all the bacteria that are making up that colony, with there are thousands of bacteria in that colony, are all the same species, which are going to be different than the bacteria on colony or bacterium 6. All of these bacteria are the exact same species, but they're very different from any of the other colonies on here. Now you'll notice that there are some places where there are some colonies that look very similar to other colonies that are on the plate, and that's because depending on where you got your sample from, there may be more than one group of colonies. But one thing we know for sure, that a colony all by itself, we can take our sterile loop and we can transfer the bacterium from colony 5 and put it on its own plate, and it will only grow that bacterium. It's a little bit more difficult if you take bacterium from the edge because bacteria 1 and bacteria 2 are very closely intermingled together. So if you were to pick it just between the two of them, you'd probably have some contamination. And we'll discuss this a lot further as we go through lab and as we go through our understanding of microbiology in subsequent weeks. So more on what causes disease. As I said before, Koch's postulates were the series of steps that he used along the scientific method to determine that specific microbes cause specific disease. So the suspected causative agent has to be found in every single case of the disease. So that's part of what his postulates are. So every single time that we see this disease, we need to be able to isolate the same bacteria. The agent or the bacteria that is suspected of causing this disease has to be grown outside of our host or outside of our patient, and it has to be isolated so that we can see for sure that it is in fact this particular bacteria. If we introduce this bacteria into a healthy host, well, usually, always, we're not going to use another human, we're going to use a lab rat or some sort of small mammal, and when we put that bacteria in that small mammal, then that host must get this same disease. And when we cultivate and when we isolate the bacterium from the mammal that we inoculated with the disease, that same bacteria should come back out again. So the same agent must be found in our diseased experimental host. So these are Koch's postulates. These are very important for you to make sure that you understand them and you remember them for your exam. All the other things that we talked about on the previous slide, on this slide here, these are just some of the steps that he went through and some of the notable contributions that he made to the field of microbiology, but it is not his exact postulates. These are more scientific methods and proof. On this slide here, these are where we have his full postulates, and you want to make sure that you understand those and you have those memorized. So other notable scientists during the golden age of microbiology, we really just spent a lot of time talking about Louis Pasteur and talked about Koch and we talked about Blücher, but we have a lot of other scientists that made some very notable discoveries. Ehrlich in 1884, he found out that he discovered E. coli, so he gave it his name, Escherichia coli, because his last name was Urschleich. And he understood that, well, he was able to determine that Escherichia coli, or E. coli, which is kind of our little microbiology lab pet, we use that quite frequently, caused traveler's diarrhea and also caused bladder infection. So more on what causes disease. Very important feature in this golden age of microbiology. Once we're able to establish that yes, microbes exist, yes, there's cellular organisms that have to come from other cellular organisms, we have to be able to see them. It's very difficult to see something unless you stain it. Now, Robert Koch, as he developed Koch's postulates and trying to figure out that specific diseases are caused by specific organisms, he used simple staining mechanism. Gram staining is a much more elaborate staining mechanism because not only does it stain the bacteria, but it goes one step further in that it stains and differentiates the two major camps of bacteria, those being gram-positive and those being gram-negative. Gram staining is one of the first steps to identifying bacteria. Hans Gram is actually the person that developed this Gram staining. We're going to gram stain a lot this semester. In any microbiology class, work this grain and salt. It's going to make you stain a lot, in the gram stain a lot. So here we have microorganisms that when we found them on the plate, when we made our smear for our slide, they were all colorless. You weren't able to determine one camp of bacteria from the other. However, when we gram stain, we're left with two different colors on our slide. Gram staining basically is going to use four reagents. You have crystal violet, iodine, which is our mordant, and then you're going to have alcohol to decolorize, and then you're going to counterstain with the saffron. So after we've finished all of the steps required for gram staining, and we'll talk about this at length throughout the semester, we have two camps of bacteria. All bacteria that we're going to discuss in this class are going to fall into two categories. It's either gram-positive or gram-negative. Gram-positives are always going to be purple, P for positive, P for purple, and gram-negatives are always going to stain red for us. Gram-neg, neg, red. It's not a true rhyme, but it's close enough. So P positive purple for gram-positives, and gram-negatives are always going to stain red. It's going to be a very important aspect of your unknown project that will happen throughout the semester. Now, what's important about figuring out what's gram-positive and gram-negative, and we'll talk about this more as well, that gram-positives are more susceptible to antibiotics than gram-negatives are. So in a clinical setting, it's very important to understand what you're working with, and as we said before, it's a very important feature or a key feature of determining or identifying bacteria. So how can we prevent infection and how can we prevent disease? Now that we know that life comes from life, now that we know that microbes also fit in that category, now that we have a further understanding that specific microbes cause specific diseases, and how can we prevent them since we know that they're around? Well, Dr. Semmelweis was a physician. He was actually an early obstetrician, and what he noticed was that when women came to the hospital, they were more likely to die as a result of puerperal fever than women that had their babies at home. And he was able to determine and figure out the relationship between the med students who would be operating on cadavers all day long, and then when a woman came in to have her child, they would only wash their hands off with water. So those microorganisms that we aren't seeing there were still present, and as the physicians were delivering the baby, they were introducing these microorganisms to the womb, to the uterus, and then these women were succumbing to puerperal fever. So his idea was to properly wash hands with calcium lime. So the lime, the acidic aspect or nature of the lime actually kills some of the microorganisms. So he was one of the first physicians, believe it or not, that advocated hand washing. You got to keep in mind that in the early 19th century, if you had to go to a doctor, that was a death sentence. So you were more likely to die as a result of the surgery than actually having the disease itself. So that's why a lot of people didn't go to the doctor, and here's one of the reasons, because they had very poor aseptic techniques. If they didn't do something as basic as hand washing, which for all of my clinicians out there that work in hospitals or in a clinical setting, you're very familiar with the foaming in and foaming out of a room, and hand washing is part of your normal daily routine. And even for those of us who are not clinicians, and washing should be a part of your normal routine. Then we had Joseph Lister, who developed this antiseptic. It was actually a phenol that was to be used in surgery, and also perfected or gave more details on how to perfect the antiseptic technique. When you look at his name, Lister, it kind of looks like another antiseptic that you might have in your bathroom. Yes, it does, Listerine. So the same formula is pretty close to what he used to sterilize surgical instruments and sterilize wounds. Florence Nightingale and nursing helped to prevent infection and diseases. She was a very big supporter, and she was very big on proper bandaging and cleaning wounds and removing shrapnel from wounds in order for them to heal properly. Snow developed the idea of infection control and is considered the father of epidemiology. Epidemiology is basically tracking a disease through populations, and he did that with typhoid fever for typhoid Mary. Edward Jenner is credited with developing the idea of vaccination. What he used is that there were milkmaids that he noticed didn't get smallpox, and one of the milkmaids told him, well, we don't get smallpox because we get cowpox from milking the cows. So what he did was that he took some of the cowpox that the milkmaids had, and he transmitted it. He infected, and this is totally frowned upon, he infected a little girl with it, and then he tried to give her smallpox, and thank God she didn't develop smallpox. So he is credited with developing this idea of immunity. But it must be stated that in the Far East, there were medicine men and women at that prior to Jenner that were doing the same thing. They would take a little bit of the scrapings from someone's skin that had a disease, they'd put it on acupuncture needles, and then they just tap it below the surface of the skin of their person that went to be vaccinated, and that would elicit immunity. So they don't get the credit for it, Jenner does, so that's why he called it a vaccine, because vacca is the Latin word for cow, and he got it from the cowpox and under the milkmaid, the whole story. Theodore Ehrlich, that we talked about before, developed the understanding of, or helped develop the understanding of chemotherapy and antibiotics. And when he had antibiotics, he thought that he had found these magic bullets that would cure or get rid of all disease and pestilence. But what we now know is that, what did the microbes do? They just evolved. So they evolved resistance to it. But he had some early work in the field of chemotherapy. So how do genes work? Microbial genetics is a little different from eukaryotic cellular genetics. Most of them only have one chromosome, but we've been able to manipulate that one chromosome and make advances in the field of molecular biology, and advances in the field of recombinant DNA technology. And we're going to actually do some recombinant DNA technology in lab where we'll make bacteria that's not supposed to glow, we'll make it glow. And then even with gene therapy, for people with severe combined immunodeficiency syndrome, we're able to insert specific genes of interest to combat that disease. So in the field of microbial genetics, Avery and McLeod determined that genes contained in molecules of DNA. Beadle and Tatum established that a gene's activity is related to its protein function. We understand very intimately now, as a result of Beadle and Tatum, that the DNA is the mastermind behind how the proteins are made in the cell. We also have been able to investigate, by looking at microbial genetics, the rates and mechanisms of genetic mutation and changes. And we can also use identify methods that cells can use to control gene expression. And we'll talk about some of those with the lac operon and the trip operon. And we'll do that in our genetics chapter. So this is just more information on microbial genetics and where we've come from and where it's taken us in this modern era of microbiology. With molecular biology, we can fully explain a lot of the function on the cell level. Dr. Pauling proposed that gene sequences could help us to understand evolutionary relationships. In fact, most phylogenetic trees, or the tree of life, if you will, they're going to be based off of genetic markers as opposed to morphology or shapes and limbs and the numbers of hairs and that kind of thing. So it was Pauling's work that helped to continue to do that. Woese and Fox determined that cells can be categorized in these 3 domains of life, that bacteria, archaea, and eukaryotes. So we kind of moved away from that 5 kingdom system where we had what we considered Monera, plants, animals, protists, and fungi. Now we can group them all into eukarya, that archaea and eukaryotes, and it was Woese and Fox that were able to do that. And they did that based on cellular structure. As a result of their work, we were able to determine that archaea and eukaryotes are actually more similar than bacteria and archaea, although archaea and bacteria are single cell, are prokaryotic. And then we also, this molecular work has been able to determine that cat scratch disease is caused by an unculturable organism, which means that we have not determined a culture method that will allow us to grow this organism in the lab without it being in its host. So with recombinant DNA technology, a lot of the things that we've been able to do is we've been able to get bacteria to produce things like insulin, clotting factors for us by taking out one of their genes and putting our gene of interest and getting the proteins out of it. As I said before, with gene therapy, we've been able to insert missing or defective genes using microbiology and using this, what we understand about microbes, and specifically viruses for gene therapy. The role that microorganisms play in the environment, as I said before, we've kind of looked at all the bad stuff that microorganisms can do, but we've looked at some of the good stuff too, that they can make clotting factors, they can make insulin, their products of fermentation can be used for various different things. Microorganisms can also act as antibiotics as well, as in the case of penicillin, which is a fungus that is a very common antibiotic that we use. There are bacteria that have a role or that can be used in the form of what we call bioremediation. In bioremediation, we use these microorganisms to detoxify polluted environments. One such use was used very recently in the Gulf of Mexico with the most recent oil spill, I believe happened in 2013, 2014, where they were using bacteria to clean up that oil spill. We also use bacteria in sewage waste treatment plants because it helps to recycle chemicals such as carbon, nitrogen, and sulfur, and it detoxifies them so that they're less toxic runoff to go into the environment. So we use them in sewage water treatment facilities. How do we defend against disease? We can use serology, immunology, and chemotherapy. You're probably most familiar with chemotherapy. It was Fleming who discovered penicillin completely by accident. Gilmage discovered sulfa drugs like sulfonamides, and they act as competitive inhibitors, and that's something that we use quite frequently. Immunology is the study of the body's defense against specific pathogens, and that really kind of falls into the category of vaccination and how we have come so far from in our field of vaccination. And we've also recognized some things that just don't quite work. If you got the flu shot last year, or if you had the flu mist last year for flu prevention, they're not going to offer the flu mist, I don't think, this year, that goes out through your nasal, that you put into your nasal column. They're going to use that because they found it to not be as effective. And then serology is the study of the blood serum, and to look at the presence of different chemicals that are in the blood that will naturally fight infection, and how we can exploit that in people or transfer those to people that are missing some of those factors or those chemicals, and to enhance their immunity. And here is Fleming's penicillin colony, and notice how we have this zone of inhibition, or areas where the bacteria, in this case Staphylococcus, probably Staphylococcus aureus, will not grow. As I said before, Fleming came to this completely by accident. He was trying to grow a lot of bacteria and had sloppy lab techniques, and he had a little colony of penicillin that was growing on there. So as a result, he was able to, and he tried it again and again, and he noticed that penicillin, this fungus, one different type of microorganism, will actually fight against another. And what does the future hold for microbiology? Well, students like you and me and everyone else that's interested in this field, we're going to keep asking more questions and keep pushing even further and further. The more questions that we answer, the more questions that we have. And one of the big questions that we have now is how can we effectively prevent the Zika virus? So there are a host of scientists and students that are working and investigating this specific problem. And in trying to answer this problem, we're going to inevitably, like Peter Fleming did, maybe just by happenstance, come upon answers to other questions that we've had or develop other techniques for the future. Bye-bye for now. See you for the next chapter.